Well, I'm interested in how organisms take cues from the environment, say changes in the environment, and transduce those cues into some sort of physiological or behavioral response. So I'm interested in the mechanisms whereby that happens, how organisms take a cue and respond in some sort of during acclimation or adaptation. I work mainly in fish, although I'm open to working in any type of organism. I mainly work in fish because they're immersed in their habitat, and so they're exposed to lots of different kinds of environmental stressors that we can explore the mechanisms of how those stressors affect the physiological responses.
So how would the oil spill affect fish? And then, just describe that, and then I'll
Right. So the oil can affect fish in many different ways. I guess the most immediate effects are going to be the toxic effects. So there are certain components of oil that are toxic, that when it gets into the organism, it affects organ function and can be directly toxic. So those are the ones that sort of immediately come to mind. But the thing to keep in mind is that fish are components of a community. So they're one member of, or a few members of a community. And so there can be oil effects on one part of that community that can then sort of indirectly affect health of the fish. For example, if the oil spill impacts or impairs the growth or the success of what the fish eat, then that could indirectly affect the fish. So you've got both direct toxic effects and then indirect effects that sort of cascade through food webs or other interactions in the community.
So how would you look at how the oil might affect fish? How would that look in your field work and in your lab?
So my lab's interest is in both the short-term physiological or toxic effects of this oil exposure, but also how those effects over a period of time could affect population longevity or population size, or actually is this exposure getting rid of certain genetic types in a population? For example, are all the sensitive individuals now gone and all that you're left with in the population are tolerant individuals? So both what are the mechanisms, what is the toxic response, if any, and also how does that toxic response affect the population and not just an individual? Because that's what we really want to get at in estimating the oil effects, or what are the effects on populations, on communities? What are the long-term health effects rather than, oh, I see a few dead fish here. What's more important is how is this affecting the longevity of the population?
So if you go out in the field, what are you going to do? If you go out in the field, what are you going to do?
So we're attacking this with two different types of tools or two different approaches. One is we're doing field studies. We're actually going into the field. We're collecting tissues from fish in different habitats along the coast, some that are going to be more affected by the oil than others. And we're also sampling each of those field types throughout a time course, both before the oil came, so we scrambled and we got out there before the oil came, and then right after oil impacted some of these habitats, and then for a long time actually we're interested in the recovery response as well. So we're taking tissues and we're measuring various endpoints. So we're monitoring a couple different things. We're getting in the field, we're taking tissues from the fish directly from these field sites at multiple sites and at multiple time points. And some of those sites and some of those time points are going to be more affected by oil than others. And we're going to be measuring biological endpoints that tell us, A, something about exposure. Have these fish been exposed to some of the chemicals that are associated with oil? And B, have there been toxic effects? So we're measuring effects in fish that are directly collected from the field. We're also bringing water from the field back to the lab and doing controlled exposures to fish embryos, because embryos, early life stages, are often more sensitive to environmental contaminants than other life stages. And effects on embryos, you might expect, would have a longer-term consequence than impairing adult health. If you're impairing reproduction, you're affecting growth, hatching success, that kind of stuff. You can imagine that that's going to have far greater implications for population health in future generations than just what's happening right now. So we're doing both the field studies, integrating those with the lab studies, and then we're also testing for whether the structure of the population from the field has changed throughout this event. So we're looking at the genetic structure of the population before and after, and basically asking, has there been a selective mortality event on certain members of the population? So my lab has particular expertise in what we call genomics tools. They're tools that we can use to look at how all the genes in the genome are turned on or turned off in response to some environmental stress. So it's a very sort of top-down approach looking at how a cell is responding to some change in the environment. So we're looking at about 6,000 or 7,000 genes and how they're turning on or turning off to make more protein or less protein associated with the response. And then, fortunately, we collaborate with lots of people because I think it's important to bring many measurement endpoints to bear on this, and nobody's a specialist in all these techniques. So I work with fish physiologists here at LSU that are looking at some tissue function endpoints. So is the liver working properly? Is there toxicity happening in the cells of liver tissues? So as far as biological effects goes, as far as physiological effects goes, we're looking at sort of the genomic gene expression response and the tissue level responses. And then we're also able to look at genetic fingerprints of individual fish and therefore the population. And so this genetic fingerprinting, like you know from a lot of forensics work, we're basically applying that to fish, and we can ask whether the genetic characteristics of a population has changed before and after. So we're doing population genetics on fish populations throughout before and after this event. So oil is actually a very complex mixture. It's one of the most complicated chemicals that you can find in the environment that's natural. I mean, oil is natural. Now, oil is made up of a lot of different types of compounds, some of which are toxic, some of which aren't. And we know a lot about toxicity of certain types of oils. So, for example, an oil spill that's been studied in detail has been the Exxon Valdez spill. Now, Exxon Valdez oil has very different chemical characteristics to Louisiana crude oil, South Louisiana crude. So we know a lot about what are some universal toxic chemicals that are in oil, that we can make predictions about which biochemical pathways are going to be turned on, which ones are going to be turned off. So we can predict certain biological pathways that are going to be turned on and turned off no matter what the source of oil. But there are going to be unique characteristics to South Louisiana crude oil that we're not sure what the effects are going to be. And so that's why we need to do this for this oil spill and not just rely on data that we might have from other oil spills to predict effects. The fish that we're working with for this study is the Gulf killifish. Local fishermen call them kakaho minnows. Local fishermen call them kakaho minnows. The scientific name is Fundulus grandis. And the reason we work on these fish is because, for one thing, they're widely distributed throughout marsh habitats all along the Gulf Coast. So wherever you find a marsh, you're going to find a killifish. Oh, the name's killifish. A kakaho. So the main reason we work on them is because this fish is distributed all on the Gulf Coast. Wherever you look, wherever you're able to find a marsh, you're going to find one of these fish. So it's good from an experimental design point of view because the spill, we didn't know where it was going to hit along the coast. So no matter what salt marsh we're looking, we're going to find this fish. So it's widely distributed and it's very abundant. It's a very important member of the marsh community. So we work with this fish for those two reasons, because for this oil spill, they're going to be there. If there's going to be oil contaminating marsh, they're going to be there or have been there. The other reason we work with them is because we got these sophisticated genomics scale tools that we can apply to this organism. And it's not trivial designing and developing these types of tools. They're not amenable to every organism. They take a fair bit of research and effort to develop these tools. So we've got these sophisticated tools designed for this fish. So we've got field sites as far west as Grand Isle, Louisiana, and as far east as Mobile Bay in Alabama. So we've got 6 field sites distributed along Gulf Coast, Gulf exposed marshes, from Louisiana to Alabama. So that's where we're sampling fish from to pull those tissues, and that's where we're also collecting the water. and we're collecting tissues from each of these field sites, from each of these time points, and then we're also collecting the water from these field sites, bring it back to the lab and doing the exposures. So 6 field sites, some of which have been more heavily oiled than others. The Grand Isle site in particular has been quite heavily oiled, so that'll be certainly a site that we're going to expect the most severe effects. Other sites, real obvious oiling of the marsh is not as apparent, but we're using a combination of environmental chemistry and remote sensing satellite imagery to estimate, to come up with an we're using these tools to come up with an integrative measure of exposure over a long period of time. Because we've just sampled these field sites 3 times over 3 months. So there could have been exposures to resident organisms within that time that we wouldn't have captured by just going and grabbing a water sample. But the biological effects could have lingered at those sites. So we want to have some sort of integrative measure of exposure, and we're using the remote sensing imagery data to try and get at that. So we're interested in both the short-term toxic effects of the oil and the long-term population-level effects of the oil. And we're using the Gulf killifish as a model for studying that in, because they're widely distributed throughout this coast. And they're important members of the community. And we know that there are populations that have been particularly hit hard by some of this oil. So really it's important to integrate those two, short-term measures of effect and long-term measures of effect, because one of the main things that we've learned from other well-studied oil spills, such as the Exxon Valdez spill, is it's not immediate lethality that is most predictive of the long-term population-level, lingering, multi-year effects that you're going to see. They're important. I mean, we all care about, not having dead birds and dead turtles washing up on the shores. But it's those non-lethal exposures that actually have a more lasting effect on populations than immediate mortality. So that's part of our interest, is estimating these non-lethal health effects and looking at how those might extend out across time to affect the population health.
How long do you think it'll take to see those effects? Is that a 6 months, 1 year, 2 years, 10 years?
Estimating exposure for this spill is going to be very difficult. There was one initial, opening of the hole in the ground. What's unusual about this spill, though, is it wasn't just some tanker running aground and all of a sudden there's oil in the ocean. It was this very continuous exposure over almost 3 months. Some of that oil took a long time to reach the beaches, and so it's been weathered out in the environment. And some areas of the coast are going to be more hit with a different kind of oil than another part of the coast because it will have weathered differently. So there was that immediate oil hitting the shore, but a lot of that oil that's on the surface is now gone. Gone where? It's a really good question. But part of the answer is that some of it is still out there in the environment. It's lying on the bottom. And it's lying on the bottom close to coastlines. And so even though this initial surface oil has disappeared, we might think that the initial exposure has disappeared. But what's really important to keep in mind is that future storm events are going to resuspend some of that oil back into the water column and re-expose some beaches and some marsh habitats. So what we're expecting is that there's going to be actually a long-term, periodic, low level of exposure. So we've actually scraped up a fair bit of the oil that's on the surface of the beach. You go take a core of oil, or you go take a core of sand, and there's oil from 1 inch down to, 10 or 12 inches in the soil. So it's actually seeped down. So also the rechurning of that sand with storm events is also going to bring oil to the surface and expose some critters. And any critters that are burrowing down into that sand or burrowing down into the sediment of marshes where some of this oil is stored are going to be continuously exposed over a long period of time. So that's what's difficult about these things, is you've got this sort of insidious hidden oil that's at least hidden from sight. It's difficult to study and difficult to predict when it's going to surface again. But we know a lot about storms along the coast, and we know that these storms disturb the environment enough that this oil is likely to reappear and re-expose a lot of these sensitive habitats over possibly a long period of time. We know oil is still toxic levels of oil are still in Prudhoe Bay, affecting some of the critters that live in the sediment up there, and that's 20 years later. People have done a reasonably good job at cleaning all the oil off the surface of the beaches, but when the oil first landed on the beach, a lot of it seeped down into the sediment, into the sand. When the oil first hit the marshes, some of it seeped down and got into the sediment and the sand. So even though people have done a good job at scraping it off the surface and getting it out of the surface in the marshes, a lot of that oil is still stored in those habitats and could be periodically resuspended by storm events that churn things up. And when those things get churned up again, then it's going to make them available to the organisms that live there again to get exposed to. So we could be facing a pretty long-term exposure to low levels of some of this contaminating oil. One of the most difficult things with dealing with such a large-scale event is integrating research efforts and integrating the availability of information across disciplines. So even though most of my colleagues are biologists, I have a lot to learn from oceanographers and environmental chemists, because I want to know what form the oil's in. Are all the toxic fractions blown off? Are they dissolved? Are they intact in what's reaching the beaches? Or what's reaching marshes? What's reaching the marshes? Where are the oils gone? What are the marsh habitats that we know have been oiled and which ones haven't? So a lot of that exposure data needs to be publicly available, or at least available to the research community, and why not make it available to everyone? So some of those data are available in the terms of summary maps from where we see oil, but actually making those geographically referenced observations available to the community, I think, is really important, both from the oceanographers and from people that were responding on the ground through BP and through the government that were taking geographically referenced observations. So making those data available to the research community would help everyone. On how we do that is difficult. It's one thing to get academics to share data. It's one thing to get academics and the government to share data between each other, especially when some samples and some observations are being guarded as closely held secrets, so they can be brought up in the court of law. So those are issues that we need to get around as a research community, or find solutions to as a research community. It's just documenting exposure. That's one of the main things. And also coordinating research efforts as far as, this is where I'm taking samples geographically, this is what I'm measuring, and these are the dates that I've been there, so that we don't have people reinventing the wheel, and so that we can actually act maybe synergistically across people, so that one group that's studying shrimps can maybe coordinate their efforts with the same sites as somebody that's measuring effects in fish or salt marsh plants. So that coordinated sharing of information to one sort of central repository, there are mechanisms for doing that, would be, I think, be important to encourage and find a solution to. I think an important point to make is that even though a lot of the surface oil is gone, and even though we're not seeing oiled birds anymore, and even though people are rebuilding their fishing industry and some of the fisheries have reopened, the critters that live in these exposed habitats and impacted habitats are nowhere near to being out of the woods yet. What we know is that these non-lethal effects are the most important things to get a handle on for predicting the long-term population consequences of this event. And those effects are only starting to emerge. The most obvious immediate events are the oiled birds and dead turtles washing up on the beach. But it's really important as a research community to mobilize the effort and to get the funding to really continue these long-term research projects. So it seems like the event is over, and it's over for a lot of people that make their living here. But they make their living on the organisms that live in a lot of these marshes, or at least the ecosystem health is what supports a lot of this economy. And the effects on ecosystem health, I don't think we've seen those yet. Those haven't emerged yet. So seeing dead birds and seeing dead turtles is heartbreaking, and is really important to mobilize us to do something. But it's those sub-lethal, non-lethal effects we really need to get a handle on to really start to understand something from this oil spill, and to really be able to get a feel for what are the ecosystem consequences of this, and how long is it going to take to recover.